Okay, are you handing over or are you doing a brief introduction?
Very brief introduction. So welcome everybody to another Fungisphere talk. Today we're very happy to have David Phalen giving a presentation. He's a wildlife veterinary expert in infectious diseases, and I will hand over to him to complete his introduction. Thank you, David.
Well, thank you for having me speak today. I've created a topic which I think would create fear in most people's minds just looking at it. But I think it'll all become obvious why I've used a different terminology in there. The fossorial marsupial we're going to talk about today is the wombat, and fossorial means that they just basically spend their time digging around in the dirt. So we have three species, two genera of wombats in Australia. We have the northern hairy-nosed wombat, which is critically endangered, and its current population is in around approximately 200 animal mark. They have two populations, the two locations where they are only found in these fenced-in enclosures, because one of the critical key threatening processes to them has been dingoes, and hybrid dogs are very good apparently at killing them. But I'm sure that land use has also played an important role in their decline. The southern hairy-nosed wombat, which you can see its distribution across the coast of the Great Bight, is also declining, but there's still estimates of an excess or near 100,000 animals left. And then the bare-nosed or common wombat, which we see locally here, is still very abundant in the places where it's found, and they've been tracking them in Tasmania, and their numbers are actually increasing. But you can see that there's been considerable range contraction that's occurring. And wombats are threatened by a number of different processes, including habitat loss, motor vehicle collisions, sarcoptic mange, and particularly in the bare-nosed wombat. And there's been very little work on other diseases that might be impacting them. And wombats, probably second only to koalas, have a really high profile amongst wildlife carers. They're highly sought after to be a lot of very educated wildlife carers that take care of these animals. And there's quite a demand for veterinary intervention in providing care for them and making sure that they survive to release. And they spend a lot of time in care. A juvenile wombat might spend up to 800 days in care, and they need to be close to 18 to 20 kilograms before they're released into the wild to survive and to compete with other wombats. So my history with wombats, I come from North America, as you all probably know. And so when I first came here, wombats were a bit foreign to me. And the first time that I was asked to do a postmortem on a wombat was back probably around 2008. And I was called on a Sunday by a wildlife carer who said that she had a wombat that was circling, that was blind, had abnormal behavior, and she was convinced that it was being poisoned by medication that people were putting out for mange. So I drove in on a Sunday, and the animal was dead on arrival. I did a postmortem on it. The following week, we had a second one, and then about a month later, we had a third from another location. And all of these had something that has absolutely nothing to do with the topic today, other than that's where I got first introduced to it. And they had Toxoplasma gondii infection, or toxoplasmosis, which in wombats causes a very specific lesion here in the brain. You can see the decolorized tissue, and it causes the blindness that we see, the dementia, and the circling. It's the result of exposure to spores that are produced, or oocysts that are produced by cats as a definitive host. And then when they get into these aberrant hosts, they can cause quite a bit of disease. In the process of doing the histopathology on these cases, I encountered something that I had not seen before, but others had. And that was the presence of these Aedes spores in the lung. And you can just see them scattered about in here. And these are fungal spores that are not replicating, that grow or enlarge somewhat in the wombat lung, and are sometimes, in some of these cases, associated with another lesion, which is this interstitial pneumonia. And at the time when I first looked at this, I was still a bit ignorant. We weren't quite sure if the interstitial pneumonia that we were seeing was actually caused by the Aedes spores, the presence of them, or whether it might be associated with the toxoplasmosis infection. But going back to the literature and other people's experience, this Aedes Sporomycosis, as it's been called, was first reported in Tasmania in 1980 in Tasmanian wombats. And you can see that the morphology of these spores is very similar to what we've just showed you, and that it's also associated with an interstitial pneumonia. Subsequently, there was a report of the same sort of lesions and the same sort of organism in the southern hairy-nosed wombat. And there was an investigation into their poor health and overall poor body condition that was reported in the southern hairy-nosed wombat. So they went out and collected a number of these animals, which were shot. They didn't see any gross lung lesions in them, but these Aedes spores were found in both adult animals and in pouch young in excess of 50% of the animals that they examined. The spherules, or the spores, were present both within cells and free in the lumen of the alveoli, and they were associated with some mild fibrosis and inflammatory cells. If you look at the histopath that they show in their slide, the lesions are quite a bit more severe than the way they describe them, and very similar to the first slide that I showed you earlier. They also talked about small granulomas in the lungs of these animals, and that usually they were associated with Aedes spores that appeared to have died. And they were surrounded by multinucleated giant cells. These are the first investigators to have actually grown this organism. And they showed that it grew in a typical Sabros dextros agar plate at between 20 and 25 degrees C, but it did not grow at 30 degrees C. And they didn't appear to have any particular problems isolating the organism. The third report that's in the literature is Sporomycosis in northern hairy-nosed wombats. And there were two cases that happened. These were animals that had adjacent territories. The first animal was beat up quite badly by the second animal that died later. And both these animals, there were real challenges with describing this disease and understanding it for a couple of reasons, but the most important one was that they died out in the wilderness, far from the pathology lab, and both bodies had to be frozen before they could be transported into Brisbane, where the postmortems were done. And so there were a lot of freeze-thaw artifacts in the lungs, but you can see, and if you look at this section of the lung here, you've got quite an asymmetrical appearance to them. And it's most likely this was a dependent lung, and that the so-called congestion that they saw was more a factor of blood settling in that lung lobe than necessarily edema and hemorrhage like they may have seen. But you can see in this section that there were large numbers of spores, many more than have been described typically in the other lung sections. And they did a trichrome stain on this, and they were able to show that there was some degree of fibrosis that was occurring in these lungs, quite possibly in reaction to the presence of these Aedes spores. The second case, so this is a first case still. And the lung lesions were not found in isolation. In fact, this animal had a lot of other problems. He'd been badly mauled by the other wombat, had severe bite wounds that were heavily infested with maggots. This animal, and this is something that we see in wombats all the time, when they're debilitated, they very commonly they're heavily parasitized. And I don't think it's the parasites, at least not the external parasites, that cause their debilitation. I think because they're sick, they're more susceptible to them. But this one had a heavy flea infestation, again, common in sick wombats. And then it had a severe a nematode infection of its digestive tract, which in and of itself could have caused it to be debilitated. So we're not really sure which came first, the chicken or the egg, whether the fungal infection in the lungs was a primary problem for this animal or was secondary in an immune suppressed animal, or whether it was just along for the ride, like we'll see happens quite commonly in other wombats, and was really an incidental finding. They were unable to grow it, but they only tried it at 30 degrees C, which is not the appropriate temperature to try to grow it. And using pan-fungal primers developed by Swirl and others, they were unable to amplify the DNA out of this lung, which is a bit strange. The second animal was the adjacent hairy-nosed wombat, the one in the adjacent territory. By the time they found that it was dead, it was already fairly severely autolytic. It had a lesion that we've not seen in any other animal with this or similar organisms. That had an exudative pleuritis, and there was a lot of exudative fluid in the thoracic cavity. And other organisms, probably bacteria, were present in that fluid and may have been the primary cause for that pleuritis. We don't know. But there were many Spores in the lung. And in both of these animals, and as well as the animals from Tasmania and the southern hairy-nosed wombat, it was believed, based on the morphology, that this organism was Emmonsia parva, which we'll go into what that means in just a minute. So the question is, are these organisms in the lungs of wombats Emmonsia parva? And what do we know about Emmonsia parva? And what do we know about another Emmonsia that historically has thought to be very, very closely related to Emmonsia parva, and that's Emmonsia crescens. We know that Emmonsia crescens grows at lower temperatures and produces very, very large spores in the tissues. They can get up to 700 micrometers across. And so based on the morphology of the ones in the wombat lungs, it would appear that they were not Emmonsia crescens, and were more likely to be Emmonsia parva, which is typically less than 500 micrometers across. And you can see in this section right here, look very similar to what we're seeing, what's been seen in other animals, very similar to what we were seeing in wombats. The problem is that Emmonsia parva can grow at 30 degrees and higher in vitro. And so we would have expected to be able to isolate it at higher temperatures. So what do we know about these two organisms? Both have been associated with disease in a large number of animals across the world. And they cause this granulomatous pneumonia in a number of different species. The species that they're commonly found in are rodents, and that's been most described in rodents across North America and Europe. But it's been found in animals in the bulk of the continents, North America, Europe, Middle East, Africa, and Asia. And since the 1960s, there's been a lesion that's been thought to be associated with Emmonsia crescens it has been associated with Emmonsia crescens based on sequencing in New Zealand in brush-tailed possums. And these brush-tailed possums are believed to have picked up this organism from stoats that have been introduced into New Zealand, and that they believe that they have probably were infected with this when they were brought into New Zealand, become established in New Zealand, and then has gotten into the introduced brush-tailed possum. In Europe, in addition to rodents, where it's very, very common, it's also found commonly in mustelids. And mustelids are otters, badgers, weevils, polecats, pine martins, and the stoat. There was a lot of conversation about how abundant it was in badgers. And I received some sections to look at from a friend of mine. And it turns out that the lesions that they were associating with Monsia and badgers are actually immature forms of the rat lungworm and the early stages of rat lungworm. So its prevalence in badgers is still a bit unknown. It has been associated with disease in humans, but that's very, very rare. And bottom line for all of these species, pretty much all of these species, except for horses, although they do have their nose down to the ground, would be that they are fossorial, which means that they spend their time, some part of their time underground or digging in the ground, or in relationship to otters and some of these other species of rodents, which are also aquatic species. So we come back to this again. So it was assumed that it was a Monsia parva, but we had these differences in the characteristics that didn't really match up. And then those darn gene jockeys got involved and they sorted it all out for us. And Martin and Felix did a study out of Spain, where they were looking at fungi that they found in fluvial sediments, runoff essentially from rivers. And they identified two thermally dimorphic fungi in Spain, and they've been named this Emmonsielloposis, how do I pronounce it? Emmonsielloposis coralliformis and Emmonsielloposis terrestris. And that doesn't seem to be very relevant to this study yet, but I'll bring that in just a minute. But if you look at their phylogram, what you see is that Emmonsia crescens lands into this clade here, and Emmonsia parva ends up into a clade that's more closely related to Blastomyces dermatitidis. And so in fact, it turns out that Emmonsia parva is a very, very distinct species, quite separate from Emmonsia crescens, and in fact is a Blastomyces. And this is some more work done by Jiang, and they found that Emmonsia crescens actually forms two separate clades, and that there's a North American clade for Emmonsia crescens and a Eurasian clade for Emmonsia crescens. And there's been subsequent studies that have shown that these different clades produce different sized adiospores in the animals that they infect. And they also went on to show that, in fact, that Emmonsia parva, which now is Blastomyces parvus, is in fact a member of the Blastomycetes and is not very closely related to Emmonsia crescens at all. And then the next study, the really quite exciting study that's been done fairly recently, is by Dancy, and they showed that different species of these Emmonsia crescens were more likely to infect aquatic mustelids and the land living mustelids. And then they showed, they got a hold of the scrolls from those two wombats that died up in Queensland, the northern hairy-nosed wombats. And they showed that in fact, that the fungus infecting those two hairy-nosed wombats was was not an Emmonsia at all, but more closely related to these Emmonsielloposis species that came out of Spain. So how does this relate to my research? So that what we have been doing and are continuing to do is to first look at the disease, characterize the disease a little bit better and the organism a little bit better in the bare-nosed wombat. So the Avian Reptile and Exotic Pet Hospital here at Camden regularly gets wombats brought into it, and often has animals brought in that either die in care or have been found dead on the road. And so our first objective was to determine whether we can grow this organism, and then we can use that for whole genome studies and other genetic studies. We wanted to see how easy it was detected in lungs and can be readily detected by PCR, using very traditional techniques, nothing special. And then we wanted to see whether it related to whether the organism that we have is the same one that was present in the northern hairy-nosed wombat, whether it was there was a cause and effect relationship between infection and the microscopic pneumonia that we were seeing, and whether we felt that there was sufficient disease being caused by this fungus that it would have a clinical significance to that animal, and whether there was an indication that it could be treated or should be treated while animals are in care prior to release. And then just overall look at the presence of this organism in captive animals versus wild animals. And the answer to whether it can be grown is absolutely. And it's very easy. You just take a piece of wombat lung that you've aseptically collected from an animal that's recently died, grind it up smash it up in a petri dish, take the homogenate, smash it up in PBS, take the homogenate, streak it out on a Sabro dextrose auger, and in about 4 days, this is what you get. This is almost a pure culture of our organism, but you can see that there are a few other fungi that you might find in wombat lungs as well. We've genetically characterized it, and we just used traditional DNA extraction methods. We've collected DNA from 15 bare-nosed wombat lungs. 6 we knew that they were histologically positive. 9 we didn't know what their infectious status was. And we found that 13 out of 15 were positive with a single amplification PCR using the primers that were used in the original study to look at the diversity in wild animals. And we've got sequence for both the ITS and the large subunit. We've subsequently collected some other sequence using other set of primers. And all these sequences are identical to the original sequence that came out of the northern hairy-nosed wombat. So it appears to be exactly the same organism. Histologically, our studies, we had 34 cases, 13 that came from the Avian Reptile Exotic Pet Hospital here in Camden, 12 that were part of the Taronga Zoo or the pathology registry collection at the Taronga Zoo, and 13 of these were bare-nosed wombats. 6 were southern hairy-nosed wombats, and 2 the species were unknown. The way we examined the lungs was that we looked at 10 fields of our cross-section of the lung under 10x magnification, and took the total number of spores that we saw over that. And then we graded the pneumonia as diffuse, which would be quite severe and possibly impacting the animal's ability to breathe to some extent. And maybe not under normal conditions, but under any stress, focal and multifocal to coalescing. And then we looked at the presence of spores versus the presence of pneumonia, and attempted to see whether there was any statistical correlation. And what we found was that the spores were very similar in size to what had been described in all the other wombats in Australia. We had the bulk of the positive animals were wild animals. There was one captive animal that had a infection, but the origin of that animal wasn't known. So it may have acquired that infection prior to being in captivity, or it might be provided with an enclosure that mimics what happens in the wild, and is allowed to do a lot more digging than the other wild wombats that are the other captive wombats that we were able to look at. We also found pneumonia in 17 of 35 animals, and the majority of the animals with pneumonia had the more severe form of the disease. And there was a strong correlation between the presence of pneumonia and fungal spores in both the bare-nosed wombats and the southern hairy-nosed wombats that we looked at. And we had 3 animals that had fungal spores but did not have pneumonia. So when we did our statistics, there was a strong correlation between the presence of spores and pneumonia, and that animals that had spores were 3.25 times more likely to have pneumonia than animals that did not. The lesions in these lungs were characterized by a marked increase in alveolar macrophages. Spores were predominantly found in the cytoplasm of alveolar macrophages, and there's this image that I have up here shows an alveolar macrophage surrounding an spore, and mild pulmonary edema and very limited fibrosis. So we don't see the chronic sort of changes that were described in that one northern hairy-nosed wombat in the animals that we've looked at in this study. So what were our conclusions? And we'll go into some discussion here, and anybody can jump in at any time during this, but the Emmonsielloposis was detected in the wombat lungs, appears to be present in the environment across the range of all 3 species. So we know that this fungus exists in southern to mid-north Queensland. We know that it extends, environmental range extends to the area of the Great Bight, and is also present and found in Tasmania. We think that the majority of wild wombats are southern hairy-nosed and bare-nosed wombats are infected with this organism based on histopathology, but it might be that with culture or PCR that we find that the prevalence is higher, and that histopathology may not be as sensitive as those other tools to find it. And disease rarely occurs in zoological collections, most likely because of the nature of the enclosures that they're in. They're not able to dig down into the dirt and create their own burrows the way they would in the wild. It clearly is a pathogen. It's clearly causing disease in these wombats. The severity of the disease is variable, I guess not unexpectedly. Life is a bell curve, and no pathogens always cause the same disease in every animal. There's always a host pathogen environment interaction that's at play. It does cause sufficient enough disease that it could impact survivability in wild or released animals. But we don't know why it causes the degree of variability that it does. In some animals, we don't see disease at all. In others, we've got quite severe disease. And then the question becomes, and perhaps this will be up for discussion after I'm finished, is whether we should or could treat these animals for this fungus when they're in care. And how would we know if treatment was successful? So in most instances, when we treat fungi, they're growing, and our antimicrobials affect a growing fungus that has active metabolism. These organisms in the lungs are not growing. They probably are metabolically pretty inert, and I don't know whether trying to treat them would be successful or not. And in humans, there's quite a bit of debate as to whether to treat them or whether treatment does any good or not as well. But they do. They treat them with amphotericin and itraconazole in people when they have this infection. They also treat them concurrently with steroids to mitigate the effect of inflammation that occurs in the lungs. But again, that's a different species. That's actually Emmonsia crescens that we're talking about, and not our species. And then how would we know how bad the disease was before we started treating, and how would we know if we got rid of it afterwards? And this might be an area where we could start looking at the possibility of doing bronchoalveolar lavages to see if we could detect the organism in them. Seems likely that we could. You can do transbronchial lung biopsies, which might be able to characterize the severity of the disease, and then subsequently treat them for a month or two, and then prior to release, go back and repeat those diagnostics, get some idea of whether the treatment was successful or not. So one of the things I was thinking about in the last few days prior to this talk is what's the relationship between this species and the wombat? Clearly, the wombat hasn't coped to deal with it entirely. It doesn't kill them generally, but it can, and it can interfere with their ability to breathe, and that can't be a good thing. So is there a possibility this is an emerging infectious disease? Could it be like Emmonsia crescens in New Zealand that has recently been introduced? I would guess for it to have this wide distribution, probably not. It probably has been here for a long time, but we don't know that for sure. It clearly doesn't benefit the wombat, but it must benefit the fungus in some way or another. And so what I was thinking about is maybe it benefits the fungus because I think a lot of these spores that we see in the lungs, in these pulmonary macrophages, are probably being carried up the respiratory elevator, being swallowed, and then probably pass through the digestive system and leave the wombat in its feces. And so it probably is a good way of disseminating the fungus around to be in a wombat. Who knows, maybe wombat poo creates a good environment for the fungus to grow once it's outside of the wombat, or maybe when wombats die, the nutrients that are released into the soil from the dead wombat may also benefit this fungus. I don't know. So that's my presentation today. I'd be really keen to get your opinion on the things that I've said, and any feedback that you think might be helpful for our research, or anything of interest that you might just like to talk about.
Thank you, David. That was extremely interesting. There's a couple of questions that have come up in the chat. I guess you can see those too, right? There's a question from David Guest about the Cox postulates and going beyond association, and a question from Tanya about how much of the disease is attributable to the organism. So why don't we start with those and have some conversation?
All right. Well, if you could write me an animal ethics protocol where I could take live fungus and infect one of these dear cuddly wombats, I guess we could fulfill Cox postulates. I think it's going to be very difficult to be able to do that, but it's potentially possible. We'd have to get joeys that were in care that were kept from exposure to this organism. We'd have to have enough animals to sacrifice at the beginning of the experiment to show the prevalence of disease. We'd have to infect them, and then we'd have to kill them. And I don't see me ever doing that and being able to treat wombats again, or walk out of my house probably. So I don't think we're going to be able to do that. We can just speculate. It does look like it was the only organism we were able to identify in the lungs. And our data, which is circumstantial, argues that it is the cause of the lesions that we see.
We have a question from Julie, or Julie's hand is raised.
David, I was just wondering, to test your theory about wombats being a good mode of dispersal of spores, have you actually tried to culture it in the feces?
No, we haven't done that yet. And it might be a little bit challenging because I suspect there's a lot of fungi in the feces of wombats. But with our PCR, I think we could amplify it readily. So that would be one of the next steps that we might try to do. They have feces from about 40 different wombats in the freezer, keeping them for a rainy day. So that might be a good place to start.
Is the organism an environmental saprophyte, or what does it do in the environment?
Well, all we know is about the other species that have been identified in river bottom sediment, and they were found in the sediment, but nobody's looked at this organism in the environment. I assume that the wombat burrow, being nice and cool, would be a good place for it to replicate and to grow. But I don't think anybody really knows what it does. Even the Emmonsielloposis species that we talked about previously, it's been hard to find them in the environment. But we have these little, as part of our sarcoptic mange research, we have these little wombat cameras that go down into wombat burrows and can collect samples and things, and robotic devices. So it might be possible that we could collect some soil samples out of a wombat burrow and see if we can grow this organism from that.
One thing I was kind of pondering as you were speaking about the pathophysiology in the wombats is how easy is it to get imaging of wombat lungs? I mean, there's some analogous to chronic lung infections in humans that we would monitor by CT scans and serological markers rather than necessarily invasive things like BALs and biopsies.
Sure. Yeah, so I think that that's definitely possible. They're a little bit more challenging to image, but with a CT, it would be a lot easier. So I think that would be another way to do that. Again, it'd be, if you saw changes in the CT pattern, you saw improvement with treatment, it would be probably still need to have some histopathological or some other way to confirm that change was actually as a result of diminishing amounts of fungus, I think.
How about the serology? Is that something that would commonly be done? Like if you can get an IgG sort of level and see how that changes over time?
Well, it certainly could be done. What kind of antigen, how would I do, what kind of antigen preparation would you recommend?
Now that I have no idea. Just thinking about how we use them clinically. But I mean, there are probably people online who know more about that than me.
Would we have to prepare the antigen against the mycelial phase, or would we have to prepare the antigen against the spores?
I'm wishing I never mentioned it. I literally have no idea. I just order the test and I know how we use them. I'm not sure if there's anybody more immunologically minded online.
Mark's there.
Can I ask a question?
Yeah.
So it's quite fascinating. Judith Nimmo sent me one of these in 2005 or something from a Victorian wombat. And I think in that one, there was almost seemingly no host response. And you've described in your big series, a couple of them having no host response really at all identifiable. So it's really interesting to me that quite a distinct variation in the host response. I was just going to follow up on something that you said, that you sort of implied that you didn't think that these fungi were growing in the lung, that they were sort of just present. Do you want to elaborate on that?
Yeah, so in fact, the definition of an adiospore, which is what is sort of a really vague term if you want to think about it, because it encompasses the spores from Emmonsia crescens, from our now Blastomyces parva or parvum, and from a number of other unrelated or in the same group of fungi, but not all that closely related. They are all believed to infect lung tissue and not replicate. So we don't see any budding like you would see with blasto or histo. They enlarge, the Emmonsielloposis crescens enlarge, and the older the animal is, the bigger the spores are in there, but they don't see any replication at all. There's no dissemination from the lungs into any other tissues. And so they're assumed not to replicate in the lungs and to be constantly exposed. These animals are constantly exposed. They're probably, I really think in these wombats, they're getting rid of them as they go along, but we don't see any replicative forms histologically. And I didn't reference your case.
It was, yeah.
I found it though.
I don't know how you would have found it.
David,
Do we know if there's, oh, sorry, David Guest. I just ask a quick one and then David Guest next. Do we know if is there any replication within the macrophages, and similar to what you'd see with Cryptococcus? I mean,
No.
I see that inside there, but
There's no evidence of that at all.
sorry about the dog. Okay, David Guest, please.
so David, what you're saying is that the the wombat picks up the spores from the soil, presumably. They go into the lung, but then there's no replication. So there's no contagion. There's no wombat to wombat infection. And so in that way, this is something I'm trying to grapple with for a lecture I'm giving next week about epidemiology. And in plants, we distinguish linear epidemics and exponential epidemics, right? So linear epidemics are when a plant gets infected, but it doesn't spread from that plant to other plants. That plant dies and the inoculum stays in the soil. So when another plant comes along, it gets infected. So you get a linear epidemic rather than, you know, your typical exponential epidemic. I'm asking because I'm grappling for an example of a linear epidemic from the animal kingdom.
Well, this would be a good example, I think. And it's if you go back, and I can send you, or I can direct you to a good paper on the studies that have been done on this. But the bottom line is that we think it's just an environmental exposure. We think that's what happens in humans as well, that the organism doesn't replicate in them, that they get the dose from being wherever they are and inhaling the dust, and that it grows in them, and it doesn't grow in them. They may distribute it, they may help disseminate it, but they don't amplify it.
I think in general, fungal infections are not spreadable. It's a dead end host. This is my understanding for most fungal infections.
Well, it's certainly true for Aspergillus in birds. I mean, it kills them, and they don't. If you have an infection in one bird, it doesn't transmit it to another bird. We may see multiple Aspergillus infections in an aviary, but the reason for that is the environment. And we've got an environment where there's lots and lots of spores, and where there's poor ventilation, and infection occurs as a result of that. So, yeah, Aspergillus is very, very common issue in birds that, really common in birds that aren't that are Arctic birds, Antarctic species, ocean species that don't encounter these spores very often. And then when you bring them into wildlife care centers on land in a humid environment, then they die from it if you don't treat them to prevent it.
Thanks for that. Great examples.
Mark Rottenberger.
I think Phalen had his hand up first. Phalen,
Oh, Mike, it's fine. Go ahead. I can ask the next one.
I was just going to ask, because it's a weird situation, isn't it, where we've got a fungus that doesn't seem to replicate in a tissue, yet we've got an association with disease. And I suppose I'm trying to grapple with that too, because I'm just thinking, you know, are these animals presumably the spores from the mycelial phase are different. I actually don't know. And are the animals exposed to lots of those spores from the mycelial phase? Is that where they're getting? Because I can't imagine that we wouldn't see other fungi if it was just present in the soil and they were inhaling soil. And I can't imagine that we wouldn't see other sort of evidence of pneumoconiosis or whatever associated with it. It's a fascinating interaction, isn't it?
Yeah, I think there's a couple of points there. First of all, if you go back, and I'm a little bit ignorant on this, but I think if you look at the original paper where they grew it in the southern hairy-nosed wombats, out of the southern hairy-nosed wombats, when you increase the temperature of the fungi, the level that they're no longer compatible with them growing, they start to form these spores. So I think that the mycelial fungi produce these spores that then get disseminated into the environment. The other thing that nobody's really talked about is the size of the spore versus its distribution in the lung and everything else. And these guys, spores may enlarge somewhat when they're in the wombat, but the typical size that we see, the smallest size we see, is about 5 microns, which my understanding is about the right size for them to be able to penetrate the respiratory system down into the alveoli. And so I think it's as much a factor of the size of the spore perhaps as anything else, so why it's able to get down into the lungs and why other fungi may not be able to.
So do you think there are mats of these mycelia growing in these burrows?
They're growing somewhere, yep.
Well, I had the same kind of problem to understand if we have just an organism which is inhaled and stays and does nothing. So how does the disease occur? Is it just a reaction towards the spores from the body to say there's an allergic reaction or something? Because normally I think you would see multiplication and dissemination and causing disease somewhere else, or making like a lesion in the lung, which then causes like the stopping of the working of the lungs. So I think for me, it's kind of hard to see if we only assume that the spores is present and does nothing, then how we associate the disease with this presence of the spores.
That's a good question, and I don't have an answer that I can base in science other than what if these pulmonary macrophages that ingest these spores die?
Mm-hmm.
And then the inflammation can be associated with necrosis, associated with the immune response trying to get rid of them. And that's the only thing that I can think of.
It can also be an allergic component similar to the ABPA and SAFs kind of picture.
Yeah, it sounds like an allergic disease, like asthma or hay fever, something like that.
You get fungal sensitization from aspergilluses, causes lung disease as well.
Phytosporiums and penicillin, all sorts of fungi, yeah.
So it might, if that was the case, what we might see is as the animal aged, that the lesions would get more severe. If we looked at the disease in joeys, the very early, their first exposure, there might be fairly limited inflammation, but in older animals, there would be more. How fast does that sensitization develop in people?
Yeah, in people, it's normally following a significant exposure, and then repeated small exposures can be triggering for asthma or for a chronic inflammatory lung condition. I suppose one thing that would be different is they'd respond to steroid therapy rather than getting worse. So it's not antifungal therapy, but anti-inflammatory therapy that is effective.
In humans, the Sporomycosis caused by different organisms is in some it's associated and it occurs in healthy people, and then in others, and there's an emerging disease that's occurring now in South Africa. it's predominantly associated with AIDS-like disease, but at least that one in South Africa, I think the organism is actually replicating in the lungs, so that would be different situation.
It's really interesting. Mark.
The other thing that strikes me as a really nice experiment to run with this organism is to look at its interaction with some sort of macrophage model, because it looks like it's got a big cell wall and lots of defenses, I suppose, but that might be part of that. And so I think the temperature thing, I like that it gets to a body temperature where it can't actually physically proliferate, but that interaction with the host macrophages is also sort of an interesting line of investigation with this one.
Yeah, you probably could purify those spores from a lung and put them into tissue culture with a macrophage, some sort of system.
Or collect them from your mycelia when you grow it.
Yeah, that's true. Heat them up.
Oh, I think we're just about out of time. So unless anybody has one kind of final burning question for David on today's presentation. No, then David, just thank you very much. It was really interesting. It was great discussion.
All right, great. I appreciate everybody's input. Give me a lot of things to think about.
Okay. All right.
Thank you.
Thanks,
All
David.
right, bye.
Appreciate it. Cheers.